Hello, everyone. I've been instructed by my producer to only introduce Nestlig. So, hi, I'm Nestlig. Nice to meet all of you.
That's
I look a
not
little
what
bit
I
different. Welcome back, everybody. Joining me today is Nestlig, one of my editors. We also have co-host. Let me try that. We also have host and co-host Peter, but he's in the background. He's running the things, making sure the creationists don't eat the wires. So that we can stay on the air. Because last week I had there were like nationwide internet problems, which was kind of wacky. So, how are you doing, Nestle?
I'm invisible, as you can see. You can't see.
You're Dan Stern Cardinal.
You can't see me. Yes.
Oh, yeah. If you guys didn't see it last time, I mean, I think everyone who's here saw it. But last time we had a show, we had Dr. Dan on of Creation Myths, and we had John Perry on of Stated Clearly, and we talked about the origin of COVID. So that was fun. It was a grand old time. Yeah, we had a good time. Nestle, did you have a good time?
Regarding what?
The COVID talk? Did you have fun doing the COVID talk?
Well, enjoy is a strong word.
Okay, fair enough. We were there. We were around.
We were there. Yeah, I also dropped a bomb on the Peaceful Science forum because I had a few things to say.
Mm-hmm. Yeah, I said that to John Perry. I don't know if he responded to you on it.
He responded, yeah, and he also I responded a few days ago, and I just noticed that he also responded today. So yeah, but okay. I won't read that right now because we are now busy doing something else.
Yeah. So the funny thing is, in the Ancestor's Tale, we have also finally made it to the photosynthesizers, the sun eaters.
Yeah.
The tail that we are currently working on, for anyone who's not aware, is the cauliflower's tail. So we are having our one and only veggie tail that we're doing.
Yeah. And fungi get a brief mentioning, and then
yeah, they get the short end of the hyphae. Yeah, they just get kind of a passing mention along with all the other non-animal unicorns, basically. The Choanoflagellates are the only ones who get their own tail, and then we jump forward or jump backwards like a billion years in history to our common ancestor with plants. So,
I think it's more like I believe it was like sometime between 1.5 and 2 billion years ago that the common ancestor was, right? Like I don't know the time divergences on top of my head. What do you think?
I mean, to the 2 billion. I guess it. Well, because we didn't really use molecular clocks are kind of all over the place for our common ancestor with Choanoflagellates, but they tend to converge around something like 900 million years ago is what I typically see. And so then, a billion years prior to that would have been like 1.9 billion. So within that range, I guess. But yeah, so the veggie tail, the cauliflower's tail is the next one, which is oddly enough about the metabolism the metabolic rates of different organisms. So that'll be interesting. And then we've got like the redwood's tail, which is about tree rings. So we'll do dendrochronology in that one. Then the humped bladderwort's tail, in which we talk about the C value paradox and the G value paradox. And I feel like there's another one, but I think I'm forgetting.
Are you breaking up or am I breaking up? I'm not sure.
Oh, am I breaking up?
Not for me.
I didn't see anything.
And it was just on my end. Then
okay.
All right.
No, I was right. So cauliflower, redwood, and then hump bladderwort, and then the mixotrick after that. So our one tail about an excavata member. And then we get 2 bacterial tails, the rhizobium and then Thermus aquaticus. And then that's it. We're done. Only 6 more videos. Nestle, we're almost there.
Yay. 4 years, I think, almost.
Yeah, we're almost at 5 years, I think. I think we're approaching 5 years. But and then we get to do inconvenient facts Al Gore doesn't want you to know. So we're going to read this book about climate change denial. Aren't you ready? Are you so excited for that, Nestle?
Yay. Yay.
Hey, you asked to be here. You're here voluntarily. Nobody can see the chain that I have, from your leg to the table. No one can see it. So what are we talking about today, Nestle?
Well, I was for a long time I was planning on to do a talk about photosynthesis, but it turns out there is too much to talk about photosynthesis in just one episode. So I just broke it up into several chapters, and we were going to talk about the first chapter today, the sun eaters. We will
All right.
we will basically cover who does photosynthesis, and also a related process called phototrophy, but I will cover that in a few moments. All right, so I think we can then begin with the slides, unless there is anything else to mention.
Not that I'm aware of, I guess.
All right.
We don't have any really anything's coming up, I guess, other than what I mentioned. If we have any guests in the near future, I guess we'll see. No creationists; they never want to come on anymore. All right, Peter, we ready for the slideshow? Oh, there it goes.
There we go. All right. And can you also make it bigger for all of us, because then I can see the slides better, just in case if I don't. Thank you. Yes. All right. So, sun eaters, the first chapter of our series, and I am slow. I'm just using my whatever free time I have to slowly work on the next chapter. So it can take a while until we do the next chapters. So we will see. All right, next slide.
I like this. This is very funny.
It's a suspense.
Yeah,
oh,
that's the top one. Like what you get in high school, then what you get in like a
college,
intro biology,
university.
and then botany. On the bottom.
Yeah. So. I saw this meme many years ago, and it's one of my favorite memes that I have.
That is a great meme.
My entire life, yeah. And it basically summarizes the entire thing that we are going to do for the next few Phylomaniacs episodes.
You know what's funny, Nestle, is I'm sorry, I did not mean to cut you off. When I was a young warthog. When I was but a wee biology student, I hated photosynthesis and cellular respiration. I absolutely hated both of them. And I firmly believe, now that I'm a professor, that the best way to learn something is to teach it to somebody else. So summarize it so that you learn it for yourself, and then be able to explain it to someone else. If you can do that, then you can get any idea, basically. Because that's how I understand photosynthesis now and respiration.
Mm-hmm. That's actually why I'm also in favor of interactive courses instead of listening. It can be fun to listen to a very good presenter. If you, for example, one example that comes to my mind is Carl Sagan. Carl Sagan can be very interesting to listen to, but most professors can be a bit boring.
Not me, of course. I would never.
My advice would be to let students talk to each other during the course, so they can learn from each other. That would be my.
I've seen. One thing I've seen, maybe you have too, is a breatharian stream. No, uncooked Matthew will not be making an appearance. One thing I saw was that you should let students. After 50 minutes of lecturing, let the students do some kind of activity to keep them engaged. Have you seen stuff like that?
Oh, sorry, I broke up for 1 second. Can you repeat that sentence?
Never again. No. I've seen stuff that says like when students like if you're lecturing to students after about 50 minutes you should let them do an activity themselves so they can try to get the material.
50 minutes or 15 minutes?
50.
50. Oh, that's actually long. I would recommend 15 minutes because I have seen that like the attention span like already begins to drop after 10 minutes for most students. So it's but in any case we can discuss how lectures are done, but although if you don't move on, we will be here until
Sorry, my bad.
3 hours. No worries. Like it's also an interesting topic to discuss. In any case, this is my overview that I have so far regarding the chapters for photosynthesis. First chapter, the sun eaters. The next chapter will concern how the impact photosynthesis had on our planet, basically. And the main impact it had was the oxygen it left behind. So, and also, if you're curious, I highly recommend Nick Lane's book, Oxygen, or how what's the full title again? How Oxygen Built the World. I think that's the title of the book.
Nick Lane,
Yeah,
oxygen,
Nick Lane,
something like that. Oxygen, the molecule that made the world.
Yes, Oxygen, the molecule that made the world.
I have not read that one. I've only read The Vital Question. I've not even read was it Transformers? Wasn't that his most recent one?
Oh yeah. I also read that one. Yeah, it's also Nick Lane's a very good book writer.
I reread a large part of The Vital Question as I was writing a lecture for my non-science biology students, because the way that he explains the origin of life stuff is really very good and intuitive. It was very, very well done.
Yeah, and it's not just like sometimes science books can be very dry in the way they are. Like they read like textbooks. You know what I mean? But Nick Lane does not read like a textbook. It reads like a novel almost, because he tells stories to explain things, and it's very, very.
Would you say he makes tales about
Yes.
about the origin of life?
Exactly. And the third chapter will harnessing light will basically go over how photosynthesis works in detail. And in the last chapter, we will discuss how photosynthesis has evolved. And it will probably be split up into multiple parts because there's so much to cover there on that topic alone. So wait and see. Stay tuned. Next slide. So just in case nobody is lost, so we can first go over some basics of how photosynthesis works. Just skipping over all the complex details, the basics of photosynthesis is you start with carbon dioxide, water, H2O, and you use energy in the form of sunlight. And photosynthesis uses those components to produce organic carbon. The details we will discuss later. But for now, we can just summarize it as organic carbon, and oxygen is left over as the waste product. Or I will say dioxygen, because it's confusing to say oxygen for both the molecule and the atomic element. So yeah, I will say dioxygen or O2 for short to refer specifically to the molecule. All right. And regarding a related process, which is what we do, oxidative respiration is basically the reverse process. It's not reverse in detail, but it is reverse in regards to the starting material and the end material. You start with organic carbon and O2, and then you end up with carbon dioxide, water, and energy again. And this energy, we use this energy to power our own bodies, basically. All right. Next slide. And as always, if there's any questions in the live chat, like I'm not following the questions in the live chat. I'm more.
Oh, I was watching it.
Yeah. So if anybody has a question, just let me know and feel free to interrupt me. or if Jackson has anything to say, feel free to.
If I have any inane comments to make, I will feel. Don't you worry.
Yeah.
Oh, I see
This
one
is
I can
also-
make already.
Oh yeah, go ahead.
Photosynthesis obviously violates the second law of thermodynamics,
Boo.
just like evolution.
Boo. Well, no, this is also something that we discussed in a previous
Oh
Phylomaniacs
my
episode.
god!
Yeah.
And you argued with that creationist we had on for like days later. I don't know how you
Right.
did it.
I haven't spoken to him in how many months? Like half a year by now.
Yeah,
I think.
you because you beat him. You probably made him realize he either doesn't know
Nah,
what he's
it's
talking
I
about.
wouldn't say I beat him, or like I don't see. Like I don't see this in terms of winning or losing. I just want to inform him, and I hope maybe he learned something. I hope, but in any case, let's move on from that. So just in short, photosynthesis uses energy in the form of low entropy energy, solar radiation, which is basically most of the light that the sun emits is in the visible range, and photosynthesis specifically also uses light within the visible range, mostly red and blue wavelengths. That's the two biggest portions that it uses, and that energy is of course used to turn carbon dioxide and water into organic carbon and O2. And the products, because of this energy conservation, the products contain free energy. There is energy contained within organic carbon and O2, which we can use to release that energy in respiration. But the energy that we give off when we do respiration, or other organisms who do this as well, that energy is in the form of a low entropy, high entropy energy. It is less useful than the energy at the beginning of the process. So eventually, that second type of energy will be dissipated as high entropy energy, mostly in the form of infrared light, which will be emitted back into space. So if you can see on the top left there, you see Earth in the infrared, and that's most of the light it will emit back into space. And important to note is the energy is exactly the amount of energy is exactly the same. The amount of energy that the Earth receives from the sun is exactly the same as the amount of energy it emits back into space. Ignoring climate change for a moment. Aside from that factor, it is exactly the same. The main difference is the entropy component, and that is why life does not violate the second law of thermodynamics. All right.
But we don't have time to unpack all of that about global warming.
Yeah, exactly. We will do that later after we finished up with the the ancestors' tales. Yeah. All right, next slide. So before we go over which organism does photosynthesis, we need to have a bird's eye view of the diversity of life. And the main difference between different forms of life, unless you count viruses as life, but we won't go into that discussion for now. So we just look at cellular life, and the main differences between those is you have prokaryotes, which lack a nucleus, and eukaryotes, which do have a nucleus. And eukaryotes tend to be much larger and much more complex compared to the prokaryotes. You can see like an average prokaryote next to an average eukaryote on this slide. And I think most people in here are familiar with that, but just in case, if anybody checks this video out who is not familiar with the background, I will just give some background so nobody gets lost as I go on. All right, next slide. So this is basically the most familiar, I think it's the most familiar tree of life that most people have seen before. You have eukaryotes on the right-hand side, and you are there, right in the animals. You also have two groups of prokaryotes, the bacteria and archaea. And it's also interesting to note that before 1977, there was only one group of prokaryotes known, the bacteria. That's the name for all prokaryotes that we used to have. But now, Carl Woese and George E. Fox discovered archaea as distinct from bacteria in 1977. And I believe they used a ribosomal RNA sequence to identify this archaea group as separate. And there's also a very distinct difference between the bacteria and archaea when it comes to their membrane. Like bacteria have phospholipids that are ether linked, while the archaea have phospholipids that are ester linked. And we have the bacteria thing. Our phospholipids are more similar to the bacteria than the archaea. But curiously, our transcriptional and translational system, the way that our genes are expressed into proteins, that is more similar to the archaea, which is one reason why archaea is closer to the eukaryotes in this phylogeny. Anything to say, Jackson?
No. No. It seems fine.
All right.
I mean,
next slide.
I do think it is quite wacky that like the difference, because Nikolai makes the comment about how like there's no selective reason for the phospholipids of archaeans and eukaryotes versus bacteria. It's like it's literally just a contingency of history.
Right. Yes. It's there's also a can of worms, but just to I really recommend reading the Nick Lane's book on the vital question. He goes specifically into that detail, and it basically he argues that the common ancestors of Luca did not have a phospholipid membrane. Basically, it may have had a precursor to both. But that's again the whole can of worms about the origins of life as well. So we will continue on. So in the previous slide, I showed that the three groups, the bacteria, archaea, and eukaryotes, are three separate groups. But things have changed a lot since that phylogeny by Carl Woese and George E. Fox was presented. So since then, we have discovered that some archaeans possessed eukaryotic signature genes, or genes that were once considered to be unique to the eukaryotes. And one group called TACK, or TAC for short, were described in 2011. And another group called the Asgard archaea were described in 2017. And then in 2020, after a 12-year-long effort to culture an Asgard archaean, that finally succeeded. And it turns out to be an obligate syntroph. So it has to coexist with another species in order to be able to grow. And this is also the reason why it was so difficult to grow it. It cannot be isolated by itself. It has to coexist with a partner, basically. And you can see one example of this on the left. You can see the archaea in orange, partnering up with a bacterium. I believe it's a bacterium. They exchange metabolites with each other. Yeah, sorry.
Which one? You said about that exchanges with a Prometheorarchaeum?
I think it's a bacterium. I don't remember.
Yeah, there's a couple
specifically.
Yeah, there's a couple of bacteria, and they all do syntrophy with each other. Hence the epithet, because they're essentially just passing metabolites all back and forth amongst themselves, and then this guy Prometheorarchaeum is part of the that's like the entangle, engulf. It's like it's 3 E's. I can't remember. It's like entangle, engulf, endogenize. There we go.
Yeah, engulf. That's one model for the origins of eukaryotes. Yes, and if you're interested in that, there is like a review paper citation that I included in the bottom right there, which goes into the many hypotheses for the origins of eukaryotes. And this is one a recent discovery that has some implication which hypothesis is the most likely one as so far as now. So but basically, most current hypotheses agree on one thing. It is that eukaryotes originated via an endosymbiosis between the archaea as a host cell and bacteria, which became the mitochondria in eukaryotes. But again, the exact details of that process is still up for debate. And again, you can see, if you want to know more, go check out that citation. All right, next slide. And this is basically in 2 slides ago, I gave one example of an old phylogenetic tree, and this is basically the new updated phylogenetic tree, where eukaryotes are no longer a separate group from archaea or bacteria. They basically are a fusion of archaea and bacteria. That's basically how it works out. All right, next slide. So now we have like a bird's eye view of the tree of life. So now we also have to go over some other terms about metabolism. So you can also classify organisms based on how they metabolize certain stuff. So one term is autotrophy, which refers to any organism that's able to feed themselves. Basically, they can convert carbon dioxide into biomass. These are the primary producers, basically. And heterotrophs are all other organisms that are not able to convert CO2 into biomass, and that's us also. We cannot do. We are heterotrophs. We have to feed on other organisms to obtain biomass. Yay! Yes.
To quote SpongeBob.
And you also have also like an in-between category of mixotrophs, where some organisms are able to be both at the same time or at different times. So yeah. But one other term is called phototrophy, which are light feeders, and they are characterized by the ability to use light as an energy source. But phototrophs can be either autotrophic, so when they are autotrophic, they use the light energy to convert CO2 into biomass, and that is photosynthesis in a strict sense of the word, when the ability to use light energy to convert CO2 into biomass. But some phototrophs can still use light energy, but they are not carbon fixing. They are not able to use the light energy to convert CO2 into biomass. So yeah, so they still are dependent on feeding on other organisms or obtaining the compounds in some other way aside from photosynthesis. All right, next slide. So you have also 2 basic categories of phototrophy based on the chemical that they use. So on the left-hand side, you have an unfamiliar type of phototrophy, which is based on retinols, and which are housed within rhodopsin proteins. And this occurs in both bacteria and archaea. But these are only able to be photoheterotrophs. They are able to capture sunlight and use that energy in some way in their metabolism, but they are not able to use that energy to convert CO2 into biomass. So they are always photoheterotrophic. On the right-hand side, that is the more familiar form of phototrophy, which uses the chlorophylls or bacteriochlorophylls. And these occur only in bacteria and in eukaryotes, which have chloroplasts.
Dun, dun, dun.
Yeah. And
could there be a connection there?
Yeah, exactly. Spoilers for never.
Oh, also, I did want to point out a comment by Smitty, who says, the symbiotic origin of eukaryotes makes it a bit questionable to call them unicellular.
Right, yeah. Basically, they are cells within cells, yes.
Yep. Exactly. So there are no eukaryotic unicells, basically.
Mm-hmm. Yeah. It's really, yeah, that's also a bit of a philosophy of biology, where what is an individual organism as well? Mm-hmm. Like, are we individual organisms? Like we have, yeah, this is also a whole can of worms.
I
to discuss.
I was reading about that. So when I was writing about what was it? I think it was when I was writing about the first Nidarian video, the jellyfish's tail. Um, we talk about siphonophores, right, and how they're
Yeah.
like colonial organisms who have specialized roles for their different parts, which is kind of unusual. And I was reading a paper about that, and then I stumbled across a philosophy of biology paper that talked about like the question of organismality in application to like, is a fetus a separate individual, or is it, you know, the same individual as the mother? And so I thought that was kind of neat. It was a very interesting paper.
Yeah.
Went down a
It's
whole
really,
rabbit hole that like
yeah.
wasted that whole morning because I was reading this paper instead of writing. But
Yeah, there's also one thing that I've seen in the book, Life Unfolding. I think you've also read that book as well, Life Unfolding. And there's like
yes,
one chapter that
great
there's
book.
like one chapter that I forget the specific developmental stage, but there's like one moment in your life where the the embryo, or maybe a later stage in the, I think it's also a later stage in the development, where where we can split into two separate people basically.
Oh, there's a couple different
Yeah,
places where you can do it. You could do it
yeah,
like
exactly.
as embryo and and a bit later, but yeah,
Yeah, but
it's
even a bit later, even when you are still, when you are like in several weeks
yeah.
into the process. There's also one common place where you can get like conjoined twins basically. Like they separate at the head, but they keep connected at the body basically. Yeah. So
Yeah,
so if it is if like basically there's one question that the author asks is like, if you have the potential to become two individuals, are you still one individual before? Like,
right.
yeah.
Yeah.
In any case, just to continue on with the slide, like uh, on the left hand side, the retinal-based phototrophy, they are only able to be photoheterotrophic, but with chlorophylls, you have both. You have both photoheterotrophs and photoautotrophs. So you are only able to perform photosynthesis in the strict sense with chlorophylls. With this. All right, next slide. And this is the example of the retinal-based phototroph. So in Archaea, there's one significant group of Archaea, which are called the halo Archaea, which live in hypersaline water, like very highly saturated water. And if you have seen like some places where you have these evaporation ponds where they basically do this to obtain the salt, they evaporate the water and the salt is left behind, and then they use the salt for commercial purposes. And very often you can see these bright red pink colors in those evaporation ponds, and those are commonly the haloarchaeans that have this pigment, which makes them this pinkish reddish color of retinols. And you can see like one example of that on the top left right there. And just to go over the mechanisms of how they are able to use the light energy to make metabolic energy. The rhodopsin that they have is basically the light energy excites the retinol compound within the rhodopsin, which is then able to pump protons across the membrane, and the protons, when they reach a high concentration on one side of the membrane, that can power the ATP synthase to make ATP. And ATP, as everybody knows, is the energy currency of life. But you need more to be able to fix carbon dioxide into biomass. So they are able to make ATP from sunlight, but they are still unable to use this to fix carbon. So they are still heterotrophs in this sense. Also, many bacteria, various marine groups have a related rhodopsin called proteorhodopsin. Well, Archaea, it's also one quirk of history. The bacteriorhodopsins in Archaea were discovered before Archaeans were recognized as a distinct group from bacteria. So So that's why they are still named bacteriorhodopsins in the archaea. In hindsight, we should have called them the archaeorhodopsins, but in any case, it's also like maybe some have wondered that whether the rhodopsins are related to our rhodopsins in our eyes, like we have also rhodopsin proteins with retinal, which we use to see the world, but they are probably unrelated to our rhodopsins. The rhodopsins that we have are not the same type of protein that of the rhodopsins that these bacteria and archaea have. All right. And any questions in the live chat about this, or Jackson?
Not really. There was a a printer asked about the Portuguese man of war, but I responded to that. No questions so far. But if you guys have any questions, feel free to put them in the live chat.
Yeah,
so we can ask
just ask away. Yeah. All right, next slide, Peter. All right, so now we turn to the chlorophyll-based phototrophy. In this case, within the bacteria, there are, as of now, 9 bacterial phyla, which are known to contain members that have this type of phototrophy. And again, this is the familiar mechanism of what plants are also using. But for now, in bacteria, you have 9 phyla. And 2 phyla of these are very recent discoveries. Like we have one phyla called Vulcani microbiota, which was discovered in 2018. And then another one was discovered in, I believe these groups were already known before, but in 2018 they discovered members which were found to possess photosynthetic genes. So it's still uncertain whether they are like autotrophic or heterotrophic, but we know that they have the potential to do some type of phototrophy. And the same is true for the other group, the Myxococota, if I pronounce it correctly. They were only discovered in 2023, very recent discovery of microbes with these genes. But the most familiar groups in bacteria are the purple bacteria, the purple sulfur bacteria and the purple non-sulfur bacteria in the phyla Proteobacteria. You also have the green non-sulfur bacteria on the second on the top right there, the green in the phyla Chlorophyll-based, and also the green sulfur bacteria in Chlorobi. And these bacteria, like some of them are able to be some of them are only heterotrophic or photoheterotrophic, and some can be photoautotrophic. But the most bacteria or most bacterial phyla are anoxygenic. They can do, even if they can do photosynthesis, they don't produce oxygen. So they instead of using water, they use compounds like sulfur or hydrogen sulfide, for example. And then they produce sulfur as a waste product instead of oxygen. So the only bacteria phyla that is able to be oxygenic for the photosynthesis are the cyanobacteria, often called the blue-green algae. They are the only ones that are able to do oxygenic photosynthesis, turning H2O into O2. And I think most people are familiar with the cyanobacteria as well. So they may not.
All my homies hate the name blue-green algae.
You're right. I'm just including the name because it's a common name. Most people are more familiar with the common name. Yeah.
You're totally right. It is really unfortunate that is like the common name that people everyone knows it by. So
yeah. So basically, algae is a term that refers to anything that does photosynthesis, except for the plants. The plants are a separate category, but of course that makes green algae a paraphyletic grouping because the plants are descendant from what we would call algae.
Well, I mean, if it's anything that's photosynthetic, it's polyphyletic then,
huh? Well.
Or you just mean among eukaryotes? Is it just eukaryotes or?
Well, in eukaryotes, like we will discuss it later. Yeah, so we will discuss how many times photosynthesis has been transferred around the eukaryotes. Yes, we will discuss it later. All right,
Yeah,
next
it's just blue green algae. Such a terrible name.
one.
Like
All right,
pineapples,
next slide.
not pine nor apples.
All right, so one thing that is often done in like biology classrooms is you make what is called a Winogradsky column, or maybe I hope I'm not mispronouncing that name because it's
Most of the students I went to school with were also Winogradskys.
Winogradsky.
I'm sorry, I'll see myself out.
But it's basically what you do is you grab some soil in the environment, you put it in a glass jar, and you often put some other stuff in it, like maybe an egg for protein and some paper as well to give it some organic matter for the bacteria to feed upon. And what you end up with after some time, you end up with these stratifications, the layers of different types of bacteria. And often on top you have the cyanobacteria, which are the oxygen-producing bacteria. But then the further down you go, you become increasingly more anoxic, so without oxygen, and you find these other types of bacteria, which are either photoheterotrophic, like for example the purple non-sulfur bacteria are the photoheterotrophic. You also see some purple sulfur bacteria, which are anoxygenic, photoallotrophic. So these are able to do photosynthesis, but they are not oxygen producing. And the same goes for the green sulfur bacteria right there as well. Mm-hmm. I think
Okay.
but I don't recommend doing this by yourself because it can be a mess and it stinks probably. I would guess if you open up the jar.
Oh yeah,
Yeah,
the anaerobic bacteria are definitely going to be smelly.
yeah. So it's like some of them, like I think it's the pulper sulfur bacteria. They are the ones that produce hydrogen sulfide, which is the smell of rotten eggs. Yeah. All right, next slide. Before there's also a cool interactive that I recommend people checking out on the bottom left by Bio-Interactive. So you can check out the details of this Winogradsky column. All right, next slide. So the cyanobacteria are by far the most abundant type of organism on the planet. But in terms of sheer numbers, like if you measure it in terms of biomass, then the plants dominate the world in terms of biomass. But if you count cells as individually by cell number, then cyanobacteria are the most population-rich organism on the planet. Like one genus specifically is Prochloroculus, which is a tiny marine cyanobacterium, which is probably the largest population size known of any genus. It has like tens of octillion individuals worldwide. That's much more than 10,000 trillion times a trillion. It's really insane how many of these bacteria are out there. And together with another abundant genus called the Synecoccus, or Synecoccus. Sometimes I really
That
struggling
sounds
to
fine to
pronounce
me.
it. Yeah. Synecoccus. Okay, all right. It's also a very abundant genus of cyanobacterium. And together, these two genera, they account for about a quarter of carbon fixation in marine ecosystems. So they are really very important ecologically as well.
Also, usually just referred to as the synproclade.
Right, exactly. All right, next slide. So now we move on to the eukaryotes, and we also need to cover some phylogeny as well over here to get the feeling of where everything belongs. So most people, we also discussed this in a previous Ancestors Tale episode as well
Yeah,
about
the
the
most recent one.
kingdoms. Yes, the most recent one. Yeah, the kingdoms of life, which you have like the 6 or the 5 classical kingdoms that is sometimes still mentioned in high schools, is Monera, which is basically the prokaryotes. You also have the kingdom of plants, the kingdom of fungi, the kingdom of animals, and also a kingdom of protists. Sometimes protists are split into the animal-like protists, protozoa, and the plant-like protists called chromista. But this approach does not work. As you can see in the phylogeny right there, you see that basically protists are all eukaryotes except for the plants and animals and fungi. But then you end up with a very miscellaneous grouping because some protists are more closely related to the animals and some are more closely related to the fungi and some are more closely related to the plants than they are to other protists. So there's a very how do you call it, a wastebasket taxon, or a very it's a the protists don't share things that are unique to them because some are more similar to the plants and some are more similar to the animals or the fungi. Yeah. So the protists, as a like I think some scientists, particularly the protistologists, will be mad at me because their entire field of study is based on this grouping. But as a taxonomic grouping, it doesn't really work, in my opinion. All right, next slide. So right now, the eukaryotes are like divided into 3 main groups, at least for now. There is still some debate on exactly the exact shape of the phylogeny of eukaryotes, but most describe the group Diaphoretics or Diaphoretices. I think it's pronounced Diaphoretics. Yeah, the Diaphoretics include the plants and many protists that are more closely related to the plants as well. And you also have the Podiata, which includes the fungi and the animals, and also the other protists that are more closely related to the fungi and animals. And you have also the excavates, which Jackson previously mentioned. But most studies that I've seen recently concluded that excavata is probably paraphyletic. So there are some excavates more closely related to the Podiata.
I wouldn't be surprised if it's even like polyphyletic. It's just they're all, and there's debates about like the extent to which phylogenies that deep are even resolvable, because like these guys are all have very, very large, very fast evolving populations, and we're trying to figure out common ancestors of things that live like upwards of 2 billion years ago, so it's like it's quite difficult at a point to figure out what the grouping is, especially if there are a lot of short branches between these different clades. So
I think it's also very, very clear that at the base of eukaryotes there's like a very adaptive radiation happening right there as well. Yeah. There are many lineages diverging around the same time.
Yeah, because that's probably that would have probably been right after like you would have gotten an initial radiation at the origin of eukarya when you have following the great oxidation event. So you have the actual like origin of eukaryotes, and you probably have a radiation thereafter. But these groups didn't extensively radiate, it seems like, during like the boring billion, quote quote. And then, as we just talked about in the previous episode, the Choanoflagellate's tail, in between the glaciation events during the cryogenian period, there was a huge explosion in terms of algae, of the eukaryotes. So that there seems to be that really big radiation happened right then.
I believe we forgot to mention that around the same time during those glaciation events, you also have like what's called the Neoproterozoic oxygenation event. Like you mentioned the first the great oxygenation event, which was around, I believe, 2.4 billion years ago, where before you had almost no oxygen, or pretty much no oxygen in the atmosphere. But then after that event, it was around like 2%, like around 2% of today compared to the 21% of today. Yeah, but then during the Neoproterozoic, oxygen steadily began to climb up to more modern levels, like above 10% and such. Yeah, that's around the same time as the glaciation events. Right. I think it's also it's also related to the diversification of the algae, because one thing is like the eukaryotic algae. Since they are bigger, they sink down faster than when they die. The eukaryotic algae sink down faster than the cyanobacteria, so that promotes burial. And when you bury organic carbon, what you end up with is the oxygen is left over in the atmosphere, and that increases the oxygen content of the atmosphere by increasing the burial rate. And I believe we will also discuss that in the next chapter of this photosynthesis series. All right, next slide. So, but eukaryotes, now we have discussed some of the phylogeny of eukaryotes, but now eukaryotes didn't inherit photosynthesis from their ancestors. They obtained it, or they acquired it from cyanobacteria as endosymbionts, which became the chloroplasts. So cyanobacteria are the original, the OG of oxygenic photosynthesis. The plants are just borrowers in this sense. All right, next slide. So I believe one of the first persons to have to propose that the chloroplasts were endosymbiotic bacteria is Konstantin Merejkovsky. And I believe you also made a video on him as well before, right? I think you mentioned Merejkovsky in one video. I think so maybe on passing chloroplast, that video, perhaps.
That might have been the case. I can look it up right now.
Yeah, it's an old video of 5 years ago, I believe.
Yeah, that would have been Hold on, I need to say it's pronounced that.
And I
don't think you did
pronouncing the name correctly. It's not Merejkovsky. It's Merejkovsky. I believe that's how you pronounce that name. Yeah.
I don't know if we did. But no, we should.
Oh, all right. But he was studying plant cells under the microscope, and he noticed that there was a peculiar similarity between the chloroplasts and the cyanobacterium. And then, of course, you had the very famously and infamously the Lynn Margulis championing her endosymbiotic theory as well in the late 19.
And also worth pointing out that we mostly don't remember Konstantin there because he did his work in Russia, and then when the Soviet Union formed, a lot of his work was kind of cut off from the West, and so Lynn does reference some of the early work done by Russian people, but a lot of that was just not accessible for a very long time. So
but also Lynn Margulis was initially very harshly rejected. On our theory.
Well, for mitochondria, chloroplast, that one caught on pretty quickly.
Oh, really?
But mitochondria. Well, from what I remember reading it, because she proposes back in what was it? Was it 67 when she proposed it, or 76? One of those. Either way,
I don't remember the exact date though.
At any rate, so by the because of guys like Konstantin there was already an inkling of an idea within among the botanical community and whatnot that there were these similarities between chloroplasts and cyanobacteria, but the mitochondria that took a lot more arguing because in part mitochondria is a lot older than chloroplasts, and it's been a lot more reduced in terms of form and the genome and so on. So, at the very least, I guess you could say of the two, her hypothesis was more the chloroplast hypothesis was more well received than the mitochondria one.
Yeah, but she also took the like in other instances, she took the endosymbiotic theory too far, because I believe she also believed that the like the eukaryotic flagellum was like a
And centrioles.
spirochete. Yeah, and centrioles as well. Yeah.
Yeah, because it was.
But those did not pan out. And she also, Lynn Margulis is also very an odd figure as well. Personally, like she was married to Carl Sagan. And she also is a pretty in other instances a pretty not a job, because she also was like an AIDS denialist as well. Yeah.
Oh, Nobel laureate syndrome.
Yeah, exactly. Yeah. So credit where credit is due. She was right on the endosymbiosis, but many of her ideas were wacko. Yeah. All right. But on the evidence, like a few types of evidence for endosymbiosis include the fact that mitochondria and chloroplasts, they reproduce by binary fission. They are not made de novo within the cell. They just reproduce like cells on their own within the eukaryotic cell. They also possess key bacterial proteins that are only found in bacteria. They also have bacterial genomes by themselves. Like mitochondria has a circular genome, and I believe chloroplasts also have circular genomes as well, which are distinctly bacterial in type. They also have their own ribosomes, because they need to be able to express their own genes. So they have their own ribosomes, which are more similar to the bacterial ribosomes than they are to the eukaryotic ribosomes as well. And the details of translation within the chloroplasts and mitochondria, are also more similar to the bacteria. Like in one particular example I have given here is that in us, the starting amino acid for all protein translation is methionine, but in bacteria and also in the chloroplast and mitochondria, the starting amino acid is N-formyl methionine, a slightly different type of methionine. So there are also many other types of evidence which very, very strongly supports the conclusion that chloroplasts and mitochondria in the cells of eukaryotes are derived from bacteria. All right, next slide. So now we begin with the one group which has obtained the chloroplasts from cyanobacterium via primary endosymbiosis. And the first group is called the Archaeplastida, or the translated, the name refers to the ancient plastids. A very aptly named group, because in their common ancestor, they received the chloroplast for the first time from cyanobacteria. And there are three main groups within this clade: the rhodophytes, or the red algae, the glaucophytes, and the Viridae plantae, or the green algae, which includes the land plants as well. So yeah, so land plants are a type, like in the cladistic sense, land plants are a type of green algae in this sense. Yes. All right. So next slide. So there are 15 about the rhodophytes, or the red algae. They are like also pretty diverse. They are over 7,000 species. Mostly are marine and multicellular. They have the common type of chlorophyll we see in cyanobacteria and also in eukaryotes is chlorophyll A. Chlorophyll A is the main type of chlorophyll that is involved in photosynthesis. But you have also these accessory pigments which are used to increase the bandwidth to capture more sunlight. And some of these accessory pigments in rhodophytes particular, they have chlorophyll D as the accessory pigment. Yeah, they also have phycobiloproteins such as phycoerythrin. Phycoerythrin is also seen in cyanobacteria, but in the rhodophytes particularly, phycoerythrin got duplicated many times. And this particular protein contains a pigment which gives them a distinctly red or brown color. Hence why the red algae are red. And some notable species that you may have be familiar with is nori in Japan, which is used to make sushi, as well as jim in Korea, and also lava bread in Wales. And also, if you are working in the lab like I am, agar is also derived from some red algae as well. And I think you're also familiar with agar as well, Jackson, and the agarose.
Yep.
Mm-hmm.
My wife teaches. One of the classes that she teaches is the microbiology lab, so she's constantly using agar.
Yeah. I'm almost making agar every single day. Almost. Sometimes not, but yeah. All right. Also interesting, there are some extremophiles within the rhodophytes. Like some of them are living in a very acidic hot springs. Like some, they are able to survive in battery acid almost. Some of these, yeah. One interesting fact about rhodophytes. All right, next slide. So now you have also the glaucophytes. And this is a minor group diversity-wise. Like we only know about 14 or 26 species, but there are probably more out there. Also, curiously, all of them are freshwater and unicellular, and they are asexual. They don't perform meiosis or sexual recombination. They are strictly asexually reproducing. And they also possess the common chlorophyll A. Plus, they also possess the phycocyanins, which is a different type of phycobilioprotein. But the phycocyanins are more bluish in color, and they lack the red pigment that I previously mentioned in the red algae. The red algae contain the phycoerythrin proteins, but the glaucophytes lack the phycoerythrin. So because they have the blue pigment, but they lack the red pigment, this makes them more green-bluish in color for this reason. And so they probably lost, like in their since cyanobacteria have phycoerythrin, and since the rhodophytes also have phycoerythrin, this means that the glaucophytes lost the phycoerythrin at some point as well. And also, curious fact is that their chloroplasts are unique in the sense that they retain the bacterial peptidoglycan. Like this is the main component in bacterial cell walls. Like in most chloroplasts, they don't have this cell wall component from bacteria, but the glaucophytes retained this component. Not very interesting. There's also another piece of evidence in favor of endosymbiosis. Like we have more evidence to confirm that the chloroplasts are bacteria, basically. All right, next slide. So now we move on to the Viridiplantae, the green algae, including land plants. This is by far the most diverse group, of course. They include the land plants, so over 500,000 species that we know of are included in this group. They are also characterized by the ability to produce starch inside their plastids. This is a common trait among all members of this group. They have chlorophyll A, the common chlorophyll, but they also possess chlorophyll B as the accessory pigment to increase the light capture ability. But curiously, they lost all phycobilioprotein. So the phycocyanin that I mentioned in the glaucophytes and the phycoerythrin that I mentioned in the rhodophytes. Viridiplantae lost all of them, all of these phycobilioproteins. So that's also one interesting fact to mention. So this, I believe I've seen some studies about this loss. Is that these phycobilioproteins are basically used to, as I mentioned before, they are accessory pigments that are used to increase the bandwidth to capture more light. But the loss, some authors have argued that the loss of these pigments allowed Viridiplantae to adapt more easily to terrestrial environments. So one thing is like you need to capture a lot of light to perform photosynthesis, of course. But when you're on dry land, then capturing too much light can be harmful. So some scientists argue that the loss of these pigments eventually allowed land plants to evolve on land more easily compared to like the rhodophytes they will do poorly on land, and that's why because of the they have this pigment, the phycoretin pigment I mentioned previously, that this allows them to capture a lot of light, even if they are far below the surface of the water. They can still capture a lot of light with these accessory proteins, but on land this becomes harmful. All right, next slide. And also one thing interesting to mention is that many times photosynthesis has been lost in several groups as well. So you have also within the Archaeplastida, you have like one genus called Rhodelphis, which is related to the rhodophytes, but they lost photosynthesis, but they still have plastids. Often when photosynthesis is lost, the chloroplasts or the remnants of chloroplasts still remain as the plastids. So they are still the endosymbionts are still there, but they are now unable to perform photosynthesis. You also have another group called the picozoans, which are also related to the rhodophytes, but these lost the plastids entirely in this case. And you also have within the land plants, photosynthesis has been lost many times, like about 15 times in land plants, and these I think always this loss of photosynthesis is associated with the evolution of parasitism. So one very famous example is the corpse lily, Rafflesia, which during its entire life cycle it exists within the tissue of its host, except for when it flowers. The only thing you will be able to see of this plant is not leaves, just a flower. That's the only thing you will be able to see from this plant. And it is unable.
Monotropa, the orchid.
Also a good example. Yeah.
Because orchids love, as Laserfan17 points out, mycoheterotrophs. Orchids,
Yeah.
I think pretty much all orchids do that, at least for part of their life.
Yeah. Also, yeah. Somebody mentioned Vileplume. Yes, this is the plant which inspired that Pokemon. Yes, Vileplume is based on Rafflesia. Oh, I will get some water. Be right back. Sorry about that.
In the meantime, we have the stylings of Benthoven, right? Right, Peter? We don't have the styling? Okay, never mind. We don't have the stylings of Benthoven.
The stylings
You'll have
of
to see Dapper
Benthoven.
for that. Yeah, Benthoven. He plays the guitar. He does his musical stylings.
Yes.
So well, we don't have it over here, so you have to go.
I have 2 guitars and a ukulele within reach, but I can't play.
I'm so sorry, Monotropa. You're correct. I'm sorry. Orchids do that, and also Monotropa is not an orchid; is in a different group. Yes, sorry, my mistake. Orchids are mycoheterotrophs, and also Monotropa does that too. Yes, my mistake.
Oh, thank you. I'm back.
Thank you, Laserfan17. Welcome back. We were talking about how you're our favorite.
Oh, I still have my earphones on, so I could hear you all the way.
Oh, okay.
So I know what you were talking about behind my back.
The musical stylings of Benthoven?
Yes. All right. Next slide. But the shenanigans with chloroplasts don't end within the Archaeplastida. You also have what's called secondary endosymbiosis, where you have a eukaryote possessing the chloroplast, which is engulfed by another eukaryote, and then the eukaryote being engulfed becomes the chloroplast of another eukaryote. So you have a cell within a cell within a cell. Cells all the way down, basically. Yeah. It's also interesting because of this serial endosymbiosis, you can end up with a chloroplast with 4 membranes, and the chloroplast can also contain a nucleomorph, basically the remnant of the nucleus of the engulfed eukaryote. But sometimes, or often, I think oftentimes, I'm not sure exactly what the frequency is, but sometimes the one of the 4 membranes can be lost, and the nucleomorph can also be lost. So you can end up with a 3-membrane chloroplast later on from this. So yeah. Oh, somebody says glaucophytes. Question mark. Well, we went over we went over the glaucophytes previously a few slides ago. So yeah,
the only group I'm aware of that has the nucleomorph is the chloro-arachniophytes, which are fun to say. I like saying that. But yeah, the weird little vestigial nucleus is fun.
Yes.
I'll put that here on the side. Chloro-arachniophytes.
Chloro-arachniophytes.
There you go. It's fun to say.
I don't know why, but that name reminds me of George R.R. Martin because of the double R in the name. Chloro-arachniophyte.
Well, maybe the chloro-arachniophytes can finish the Winds of Winter, you know?
All right, next slide. Speaking of which, there they are, the chloro-arachniophytes.
Da, 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 da.
Yeah, so 2 examples of other eukaryotes which obtained the secondary endosymbiosis, or yeah, so you have primary endosymbiosis with regard to the Archaeplastida, but then secondary endosymbiosis. 2 examples are the discobids, or one example is discobids, specifically the euglenids. These have 3 membrane plastids derived from green algae. And they also have, like, curiously, if you see in the image right there, they have like these red spots. They are eye spots, which they use to detect the direction of light. Basically a very simple form of eye, almost. Yeah. This is like one example of why it is sometimes very difficult to define exact terms in biology, because now, like, how do you define an eye? When you see a case like this, is this a bona fide eye or not?
That's a fun one. Yeah. At the aquarium where I work, because when you tell guests about the moon jellyfish, for instance, you say, well, they can see the light, and they'll say like, oh, well, do they have eyes? It's like, well, how do you define eye? Is an eye any sight structure? If so, then yeah. I don't know.
Also, if I'm not mistaken, the red spot that you see right there is not the eye itself. It's basically used as to create a shadow, and the shadow is used to detect the direction of light. So yeah, I believe that's how the red spot works. Yeah. All right. So, and then the next group is the chloro-arachniophytes, which are mixotrophs. So they can do both photosynthesis, and they also consume other organisms as well. So they are both autotrophs and heterotrophs. And they have 4, as you mentioned before, they have 4 membrane plastids with the nucleomorphs as well. And interestingly, the plastids that they have are from a different group of green algae compared to the aforementioned euglenids. Next slide. Oh, somebody mentioned the eye spots contain carotenes, if I remember correctly. I'm not familiar with that, but I wouldn't be surprised, because red pigments are almost always carotenoids, yeah, or carotenes specifically, yeah, or at least, yeah, or at least I think there are also some chlorophylls that are red in color, but they are not found within the eukaryotes. They're more common in the bacteria. Yeah. But in any case, the next group, we have also many other groups which have secondary chloroplasts, and these are from the so-called chromists, the plant-like protists, and they have plastids derived from the rhodophytes, the red algae, and most have chlorophyll A, the common form of chlorophyll, and they have also the accessory pigment chlorophyll C, which is unique to these groups. And so you have four groups in total. You have the cryptomonads, like from, if you see on the phylogeny right there, I'm going bottom clockwise on the bottom left, you have the cryptomonads, then you have the haptophytes, then the mysozoans, and then the ochrophytes. And we will go over a few examples within each group. All right, next slide. So the cryptomonads, they are relatively speaking a minor group. They have only about 230 species identified. They are marine, freshwater, and also brackish water. Some are mixotrophs, and they also have 4 membranes and a nucleomorph as well. So they are similar to the chloro-arachniophytes as well. So so there are other groups that have the nucleomorph as well. Turns out. Our next slide. Next is the haptophytes, and these and most haptophytes are the most famous members are the coccolithophores. One thing to previously mention is that they also possess the accessory pigment called fucoxanthin, which is a carotenoid, if I'm not mistaken. Yeah, it's not a carotene, but it is a similar compound compared to the carotene, the fucoxanthin. And coccolithophores are a very famous group of eukaryotes. They have like shells made of carbon or calcium carbonate. They account for a very significant portion of the marine productivity, like about 1 to 10% of the carbon fixed within marine ecosystems are performed by the coccolithophores. And sometimes the productivity can go up to 40% when they undergo algae blooms. And also, very famously, their fossil deposits forms these chalk deposits. And most famously, you have the White Cliffs of Dover. It's a very famous chalk deposit, which are basically billions of these, maybe even trillions. I wouldn't be surprised if there were like trillions of these individual coccolithophores just building this entire mountainside almost. And they are also the reason why the Cretaceous is named that way. The Cretaceous means chalk, the chalk era. And it's also, if you are Dutch, then it's more literal because we just call it chalk in our language, the Cretaceous. And Peter will confirm as well.
Yep, calc.
Yes, chalk. Just chalk. All right. And also interesting to mention is that when the coccolithophores die, their shells sink down into the ocean, and about 35% of the ocean floor is covered by these coccolithophores. And in some places, this can be kilometers thick. This ooze that they left behind. So yeah, very significant. Next slide. So now we move on to the next group, the mesozoans, and there are several different groups within them. But I will only mention a few of them. Otherwise, we'll be here forever. The two most famous groups are one of the most famous groups within this mesozoans are the dinoflagellates. About over 2,200 species of dinoflagellates are known to us. They are a significant part of the plankton community, and some of them are also bioluminescent. I think if you have ever been to like tropical seas and stared outwards into the sea at night, and you see the waves breaking at the shore, sometimes you can see these flashes of blue.
Or to the Netherlands 2 weeks ago.
Really? Yeah.
Yes.
Right?
Yes.
Also, oh my God. That's not a good sign.
We had
not a good sign.
I've seen several photographs. It was and videos. It is insanely pretty.
But that's not a good sign because they are only found in a very tropical seas, and when so they and it was very hot in the Netherlands too. Yeah, that makes sense. Yeah.
I'll post a video in chat.
Mm-hmm. But I've seen this when I was like in the Caribbeans. I've seen those blue flashes of light. Yes, and what basically happens is, I believe they use a pigment related to the chlorophylls to produce these flashes of light. And the reason, like one reason why, is not exactly clear, but the most common explanation I've seen is that they basically produce this blue light to alert predators. So basically, dinoflagellates are preyed upon by plankton-eating fish. But when plankton-eating fish disturb them, then the dinoflagellates will basically say like, "Oh, blue light. Here is a bigger fish. There is a fish over here. Please help us." That's basically what they are doing, or at least that's the most common explanation that I have seen. They will alert the bigger fish to eat their own predators that they are afraid of. And also another group of the mesozoans are the apicomplexans, and they lost photosynthesis. They still retain the plastids, and their plastids are named the apicoclasts specifically, but they are not able to do photosynthesis. And most of them, also one interesting fact is that because they still have plastids, that makes them vulnerable to certain types of herbicides. So, yeah. And also one famous or infamous member of this group is the Plasmodium parasite, which causes malaria. And I've seen some studies which looks into the fact that since they are vulnerable to herbicides, maybe there's like one source of a new drug that we can use to treat malaria with that. So one interesting case of how evolutionary biology can contribute to a new drug discovery. Mm-hmm. All right, next slide. So then the last group is the ochrophytes, and again there are many groups within them, but I will only mention two of them, the two most significant groups that you probably are aware of. Like one of them is the one thing to mention about the entire group, the ochrophytes, they have the pigment fucoxanthin, the same pigment that I mentioned before regarding hold on, I think that's what the haptophytes. The haptophytes, they contain also the fucoxanthin, and the ochrophytes as well. And this pigment makes them look gold and brown. And that is also one reason why one famous group of this, the Phyophyceae, or better known as the brown algae, hence why they are named that way, because of the color that they have because of this pigment. And there are about 1,500 to 2,000 species of these. And significantly, the kelp forests, which are included in the brown algae, they cover 25% of coastlines around the world, but they are mostly restricted to the temperate and Arctic regions, where temperatures don't get too cold or they don't get too hot. Basically non-tropical waters is where the kelp forests thrive, and because of that reason, they are very vulnerable to climate change. When the ocean gets too hot, then they don't do well. All right, next slide. And I believe somebody already mentioned them, the diatoms. Yes, they are also my favorite type of ochrophytes. Diatoms, they are incredibly diverse. I have seen that there are only 12,000 species identified, but people estimate that there are between 100 and 200,000 species estimated to be out there. They are incredibly diverse.
That's a few.
And they are unique because they construct these frustules made of silica, basically glass. They build glass houses. That has often been said. Cells within glass houses. And they are not only diverse, they are also the most abundant phytoplankton in the ocean right now. Like about close to half of the primary productivity within the oceans are due to the diatoms. And,
well, they are definitely not throwing stones.
No.
Mm.
And about like 1/5 of every breath of oxygen you take comes from them as well. Yeah.
So who even needs trees? That's what I always say.
All right, next slide. So the question is exactly how do these 4 groups obtain the chloroplast from the rhodophytes? Well, there are 2 main hypotheses. The first one is called the chrome alveolate hypothesis, which claims that there is one single secondary endosymbiosis event in the common ancestor of these 4 groups. Next slide. But the problem is that if this is true, then the plastids had to have been lost in these other groups many times independently. And if I, as far as I'm aware of, the other groups also lack plastids entirely. So not only must photosynthesis have to be lost, the plastids must have also been lost many times in order for this hypothesis to hold up, basically.
Nestle?
Oh, yes?
So I looked it up. The one that we had here was Noctiluca scintillans, and that's a marine species of dinoflagellate that can exist in a green or a red form, and it is found worldwide, so not just in tropical waters.
Ah, all right.
And it gives the water blue.
But they will only produce this blue. They only produce this blue pigment when they are like a lot of them, like when they are blooming, right?
Yes, yes.
So,
and we see them mostly when the weather has been warm for a little bit, which we've had.
Yeah. It was like very tropical, warm in the Netherlands recently as well. Yeah.
Yes, and it's again tomorrow.
Oh. All right. Next slide. These many losses of the plastids are it's one problem with this hypothesis. All right, next slide. And another problem is the phylogeny. So most phylogenic studies conclude that the cryptophytes are sister to the Archaeplastids. So if this hypothesis is true, then that would imply that the ancestors to the Archaeplastids and also the ancestors to the rhodophytes once had rhodophyte-derived plastids. Of course, that's a chronological paradox that it is impossible to happen. So there's also one significant reason why this hypothesis is not very favored. All right, next slide. So an alternative hypothesis is called the rhodoplex hypothesis, where you have a series of transfers from one group to another group to another group. And there are 2 versions of the rhodoplex hypothesis. So one version you can see right here is that the first transfer occurred from the rhodophytes to the cryptophytes, or the cryptomonads specifically. And then you have the cryptomonads transferred to them, and then the cryptomonads were engulfed by the haptists and also independently by the ochrophytes. And then the haptists were engulfed by the mesozoans in this particular order of transfers. All right, next slide. With a more favorable order is this one, where you have the first transfer occurred to the cryptomonads, and then the cryptomonads were engulfed by the ochrophytes, and the ochrophytes were engulfed, or some ochrophytes were engulfed by the mesozoans and the haptists. And this transfer order is more supported based on the details of the photosynthesis that we observe in these groups. Like, for example, the fact that the ochrophytes and the haptophytes share this pigment, the fucoxanthin pigment. That is more consistent with this particular order of transfers. So yeah, it can get very complicated when you consider how many times the plastids has been transferred again and again and again. Mm-hmm.
Would you say that there's been a lot of passing chloroplasts?
Exactly. Yeah.
and that some have maybe made a video about this.
All right, next slide. All right, so furthermore, it goes on still. Within the dinoflagellates, they have plastids, although half of them have lost photosynthesis, but they all have the plastids still. But not all of them retained the original plastids of the Mesozoan ancestors. Again, the Mesozoans are the larger group, and the dinoflagellates are part of the Mesozoans. But most, or about, or maybe I see. I don't know what proportion of them have lost the original plastids, but some of them, at least some of them, have replaced the original plastid with a new plastid. Next slide. So you have like one genus called the Lepidodinium, and also two strains called TGD and MGD. They possess green algae-derived plastids, and this replacement is called serial secondary endosymbiosis. Serial because they have replaced the first plastid with the secondary or serial plastid as well. All right. And also one interesting thing is that you can see right there in the graphical image, you can see the nucleomorph within the plastid right there, labeled N. The P is a plastid and the N is the nucleomorph within the graphical representation of Lepidodinium. All right, next slide. And then another group within the dinoflagellates, they have replaced a plastid with the plastids derived from the haptophytes. So this is an instance of a serial quinary endosymbiosis, because the haptophytes are an instance of quaternary symbiosis, because you have like 4 transfers, like one transfer from cyanobacteria, and then then 3 more transfers within the eukaryotes until you end up within the haptophytes, and then another transfer from the haptophytes to the dinoflagellates, ending up with a quinary endosymbiosis event. 5 transfers. Whack, yes. And you can also see, let's see here. So one interesting fact about them is that these groups are also responsible for some harmful algae blooms. They are also often called red tides in common vernacular. And they produce, like when these blooms happen, they produce neurotoxins. So yeah, be careful when you this gets mentioned on the news, because it can you don't want to be poisoned by these dinoflagellates. Mm-hmm. All right, next slide. And then yet another group of dinoflagellates, they replace their plastid with a new plastid derived from diatoms. And for this reason, these groups are often called dinotoms, because dinoflagellates and diatoms, like concatenating the names, you get dinotoms. Mm-hmm. I will give this a pass. I think this sounds a lot better than the stupid name Archaeplastida. Yeah.
Fair.
Yeah, fair. Yeah, this is a much better name. Concatenation, and in this case it makes sense compared to the Archaeplastida. In this case, it actually makes sense to concatenate the names to make this one. All right, and so they also retain in this case, also interesting fact is they retain a nucleomorph, but in this case the nucleomorph is still large, and you also have minimal gene loss, and the plastid still has like an endoplastic reticulum, and also its own mitochondria as well. So it's almost like there is a whole dinotom within the dinoflagellate. It's almost intact within it, basically.
Mm-hmm.
And you also see one, like you can see the red eye spot labeled ES in the graphical representation right there, the ES, that red eye spot is probably the remnant of the old plastid. That one. Mm-hmm. Yeah. Oh, did you want to say something, Jackson?
No, I didn't say anything.
All right, next slide. I heard you say something, or maybe just like that.
Curious.
Yes. Also, I mentioned the primary endosymbiosis in the Archaeplastida, but it's not the only time when cyanobacteria has been transferred into the eukaryotes. There's also another independent primary endosymbiosis within a genus called Polynella, within the eucalyphids.
And theirs is like a Senecacoccus relative instead of the Archaeplastid chloroplast is the closest relative of Glio margarita. So
yeah, it's not only an independent event, but also a very different group of cyanobacteria as well. Yeah, and it's also a recent acquisition. Like Archaeplastida probably obtained their chloroplast like over 1.5 billion years ago, but Polynella they got them around the Cretaceous period, so a very recent, comparatively speaking, a very recent acquisition.
like it was yesterday.
Yeah. And yeah, also interesting to mention is it's like other groups, like some diatoms and some haptophytes also have cyanobacterial endosymbionts, but these don't photosynthesize. They fix nitrogen for them. So they are nitrogen fixing endosymbionts. All right, next slide. And furthermore, you also have compared to the permanent endosymbiont previously mentioned, you also have like transient endosymbiosis, which is often called kleptoplasty. So what eukaryotes often do is they eat other cells by engulfing them and digesting them inside. But kleptoplasty occurs when when an algae is ingested, but it's not fully digested within the cell. The chloroplast stays intact for some time, and that can provide some nourishment to the kleptoplast, or to the one obtaining the kleptoplast. Yes, and the kleptoplasts are different groups retain the kleptoplast for different times. Some retain them only for a few days, but sometimes they are retained for years at an end. So kleptoplast is also a plausible precursor to the endosymbiosis. So basically, in kleptoplasts, the plastids are eventually lost because the kleptoplasts don't divide at the same rate as the host cell. But then, in order to establish permanent symbiosis, all you need to do is to retain the plastid indefinitely by having them divide in a synchronous manner compared to the whole cell. So it's a very intuitive precursor to how you go from kleptoplasty to permanent endosymbiosis. All right, next slide. And I believe this is also something that Dr. Dan has mentioned on his channel before. How we still observe this evolution happening today as well. Like one particular example of this is in euglenids. I mentioned euglenids before. Most euglenids have a permanent endosymbiont, but there's one genus called Rapaza, which feeds exclusively on the green algae from the genus Tretacelnis. And the kleptoplast, when they obtain the kleptoplasts from this genus, the plastids changes shape such that it becomes more similar to the permanent chloroplasts of other euglenids. And the rapaz is able to retain these kleptoplasts for up to 35 days and survive with them. So it's basically a transitional species that we still have, showing the intermediate condition between permanent endosymbiosis and transient endosymbiosis. All right, next slide. And kleptoplasty occurs, again, one example is in euglenids with Rapaza. You also have kleptoplasty occurring in another group called the centrohelids, which steals the chloroplasts from the ochrophytes. And another group is the cataplevarids as well, and they steal them from green algae.
I think this show should just be making Nestle pronounce like increasingly difficult words.
Yes.
We're just gonna come back with a list next week and be like iridocyclitis, you know.
And at the end of the talk, my tongue falls on the floor. Oh, next slide. And another famous group, the foraminiferans, or they are often called the forams for short.
Yeah, forams.
Yes, and they also do this kleptoplasty from the ochrophytes as well. And they are very diverse, like about 10,000 species living today. And we also know about 40,000 species that are extinct. And they are a very old group. They go all the way back to even before the Cambrian as well, or just before the Cambrian. I believe they appeared just before the Cambrian too. They also have shells made of calcium carbonate with various shapes. And these different shapes makes them useful as index fossils because they are very unique. So some species or some genus are very unique to a particular time period. And that's why we use them as index fossils as well. And some of these forams, they get the kleptoplasts from diatoms. And they can retain these plastids for days to months at a time as well. Next slide. And then you have also the ciliates, another group, which gets kleptoplasts from a wide variety of sources. Like there's some of them from ochrophytes, some of them from the hepatists or the cryptomonads. But most of them retain them only for a few days, and they are unable to solely survive on photosynthesis. So this is more of a how do you call this? A supplementary source. So they are not able to only survive on photosynthesis. They still have to go out and eat other organisms while they are still carrying the kleptoplasts. But if one genus called Mesodinium, they exclusively get the kleptoplasts from cryptomonads, and they retain the nucleus as kleptokaryons with half-life of about 10 days. And these are able to be fully autotrophic for 4 generations. So there's one exception to this rule. So the one genus is able to survive on photosynthesis alone when they have obtained these kleptoplasts. Next slide. And sorry if I hope I'm still being clear because my voice is beginning to deteriorate at the moment. Am I still audible?
Yeah, we hear you.
All right. I don't know why. After an hour of talking, my voice gets pretty bad, and I don't know why. Maybe it's because the way I'm speaking is really bad for my voice. I don't know. All right. So going back to the dinoflagellates again, they are very specialized how do you call the term of
kleptoplastids?
They are very specialized into stealing plastids. They really
obligate kleptoplasts.
Obligate almost, yeah. Mm-hmm. I don't know. And this phenomenon is ubiquitous among this group. Let's just say that. And most get get their kleptoplasts from the cryptophytes, but in total they can get from other sources as well. Even cyanobacteria, some of them, in this case, they don't steal the plastids from the cyanobacterium, because cyanobacterium don't have plastids, of course, but they just retain the cyanobacterium for some time within their cells. In this case, for the dinoflagellates. And one genus, the Phylomaniacs, they maintain the haploid kleptoplast for 30 months. And they are also unable to survive without photosynthesis. So yes, these are obligate kleptophagus. Is that a term? Kleptophagus? I don't know.
Okay.
I don't know if that is the term to use, kleptophagus. So they are obligately they have to steal the plastids in order to survive. They cannot survive without them. It's pretty weird when you have to steal plastids. So maybe they will eventually transition to permanent endosymbiosis, and then they don't have to depend on stealing them anymore. There's also one possibility. And also, one fun fact: you have also an instance of second-hand kleptoplasty. So we have one genus of dinoflagellate, the Dinophysis. They steal plastids from a ciliate, Mesodinium, which stole its plastids from the cryptophytes. So Junk, I get this plastid from you. No, I will take it yours. Thank you. All right. It's also pretty wacky as well. Next slide. All right, so we are almost done. We also have a few examples of kleptoplasty among the animals. So we have some flatworms, which get their kleptoplast from the diatoms. You also have the nudibranchs, and the famous, probably the most famous example of kleptoplasty among the animals are the cycloglossum sea slugs. Yeah, and I believe you also mentioned them as well in a few videos too.
We did, yeah, in Passing Chloroplast. Yeah, we talked about that.
Yeah, they get their kleptoplast from green algae, and others like the Elysia genus, they take them from the ochrophytes. And these kleptoplasts, they can last up to 10 months inside specialized cells lining the gut of these sea slugs. All right, next slide. So I think this is the final slide. And one last thing to mention is you also have
second to last.
In addition,
second to last.
Second to last. Sorry. Mm-hmm. So in addition to the endosymbiosis and the kleptoplasty, you also have the option of just closely hanging out with a neighbor that is able to do photosynthesis. So this is an example of photosymbiosis. You are living like one non-photosynthetic organism is living alongside one that does do photosynthesis. And so they can share, in this case, they can still share nutrients between them. So in this case, various protists have or do photosymbiosis, like ciliates and centrohelids, and also forams. In addition to kleptoplasty, they also do photosymbiosis as well. And among the fungi, the famous case in fungi are the lichens. In this case, you have one or more fungal species, the mycobionts, coexisting with one or more photobionts, which are often green algae as well. And I believe sometimes there are also cyanobacteria within these lichens too. But most commonly they are green algae. So lichens are not one thing. They are fungi and algae in one thing. Yeah. And it's also another good example of how difficult it is to define a single organism. Is our lichens a single organism, or are they multiple organisms working together? Yeah.
Philosophy of biology at it again.
Philosophy of biology again, yes. And also, you have many sponges. Can also have symbionts. They have cyanobacteria or dinoflagellates or rhodophytes and green algae. They can coexist in the sponges to provide nutrients. There is also in cnidarians, like there are some jellyfish and hydras and sea anemones as well. But most prominent example of cnidarians, which we recently mentioned, are the corals, which have symbionts from dinoflagellates, specifically the genus Symbiodinium. Is the most common. I believe is it the only genus, or I think it's the only genus that does this in the corals. Do you know?
That's the only genus I've read about. I believe it
Yeah.
is the only one. I mean, there are lots of different species of them, especially in the like Australasia area. There are a bunch of different species, but yeah, that's the only genus that I know of.
It's probably if not the only genus, it's probably the most common.
Sure.
Photobiont within the corals. Yes, and that's the reason why the corals. I think most people can make the mistake that corals are like plants. I think it's like a common misconception. But corals are animals, but they do have these photosymbionts that allow them to do photosynthesis, and they are a very important aspect to tropical marine ecosystems.
And as we talked about in the Polypiferous tale, anemones, which are animals, are named after the flowers, even though they kind of look like flowers.
Yes.
Because why not? Why not?
And there are also a few cases of photosymbiosis within the bilaterians. So you have a few flatworms that also do this. And there's also one particular subfamily of bivalves called the Tridacninae. And I include one picture of the bivalve over there.
That's the genus that the giant clam is in.
Yes. That bright blue light, those are probably like one reason for that bright blue light is probably the not the endosymbiotic, but just photosymbiotic algae existing within the tissue. Yeah. You also have a few sea squirts or tunicates that also have photosymbionts. There's also an interesting case of amphibian embryos, which have these algae living on and inside the embryo, or maybe just outside on the egg, perhaps. I'm not sure exactly the details, but you can see like one picture of the embryo right there, just with the algae surrounding or just being attached to the egg with the embryo inside. And I think, and I would suspect that the relationship is this, because basically within the embryo there is like a source of protein that nourishes the developing embryo, and then the waste it produces is basically ammonia or urea, basically, and that can be used by the algae as a source of nitrogen, and then the algae can in turn supply a carbon source for the embryo. So this is one example of a mutual beneficial coexistence between a photobiont and its host. All right. Our next slide. So this is basically the slide summarizing all photosynthesis that occurs among the eukaryotes. Eukaryotes don't do photosynthesis by themselves. They have to have either endosymbionts, or they have to have either kleptoplasts, or they must coexist with a photosymbiont to do photosynthesis. All right, and that's basically it for today. Next slide. And then for the next chapter in the series, we will discuss how oxygen has changed everything on our planet. All right. And
Yay!
4 minutes within time. Yay.
Woo! You did it. Woo. There you go. I didn't realize my camera was down. Yay! We did it. We talked about the diversity of the Sun Eaters.
Mm-hmm.
Do we have any questions from the side
we
chat?
still have a few minutes until we are forced.
Yeah, we did
the Time Lords. We are forced by the Time Lords to end this stream.
I really enjoyed when we did passing chloroplast. I thought that one was a lot of fun.
Mm-hmm.
That video. I think I was reading Entangled Life at the time, which was in part where I got the idea, which was by Merlin Sheldrake. If you guys haven't read it, it's quite good. It's all about fungi, and he has a whole chapter on lichens. So it's highly recommended. Now it's not going to go back in. It never does. Oh, yeah. Wait, there it goes.
And I've also watched a lecture on somebody who specializes on lichens, and it's very surprising how difficult it is to grow a lichen in a lab. Basically you have to starve the fungi in order to coax them to become a lichen. Because if you give them too much nutrients, then they go, well, I don't need to become a lichen. I can just hang out on my own.
That's I wore the perfect shirt for this. See, my got algae shirt. And then on the back, I don't know if you guys can see it. Can you guys?
Got algae, freshly salted.
Did you see it?
Yeah.
All the different tangs.
All the different tangs.
Because I got this shirt at Aquashella in Dallas last year, which is a big aquarium, home aquarium convention, and I saw them selling this shirt, and I was like, I have to have that. It has to be mine. Mm-hmm. So, yeah.
Also, Smitty asked, have chloroplast or mitochondria ever become independent again? No, I don't think they have. No, they are.
Also, in one sentence, explain the origin of cell nuclei.
Read Nick Lane's book on the vital question. That's he discusses the nucleus. It's in a few sentences. It basically relates to how in eukaryotes you have introns, which have to be spliced out before they can be translated by the ribosome, and the process of splicing is slow compared to the process of translation. So that's why the splicing has to be kept separate from translation. And in eukaryotes, splicing occurs within the nucleus and translation occurs outside the nucleus. And that's why that the structure exists. How that exactly originated, again, there's a whole can of worms. Yeah.
Yeah.
So I would if you we mentioned the end goal, the triple E hypothesis, the EEE. What was the full name again?
Entangle, engulf, endogenize.
Yes, that model is actually one of my favorite models because it also explains not only the nucleus but also the Golgi apparatus and the endoplasmic reticulum. Basically, these membrane-bound organelles, they originated by first having these protrusions going out and then surrounding the whole cell like that. And that's how the structure originated in a very offhand way. Of course, the details are very complicated to go over. Yeah.
Prometheorarchaeum is whack.
Any other questions, perhaps, or maybe I'm.
Introns originate as remains of transposons. I think I've heard that too.
Yeah. They are. I believe they are type 2. What's the full name again? Let's see. Yes, they are like in bacteria. There's a group of like they are called group 2 introns, and they are self-splicing, and that's probably the precursor to our introns that we have.
So for instance, Smitty, the endoplasmic reticulum is like continuous with the nucleus. So when you do transcription translation, right, you take your DNA, you transcribe it into an mRNA sequence, and then it leaves the nucleus. And then once it is outside of the nucleus, it attaches to a ribosome on the rough endoplasmic reticulum. And so then that sequence, that mRNA sequence, is then translated into an amino acid sequence that then gets folded and packaged, and that folding and packaging and all that sort of stuff that happens in the Golgi apparatus, and then it gets sent off to wherever it needs to go in the membrane or what have you. So they're both related, right? They both involve or are involved in transcription, translation, and post translational processing and all that sort of stuff. So, all right. Well, it is a little bit past 9 my time now. So, thank you, Neslig, for that presentation, and then we will continue with that next Thursday. And thank you, Peter, for hosting, and for everybody else, thank you guys for watching, and then we will see all of you next time. So, bye, everybody.